All right, class. So wrapping up our cardiac cycle. So as we understand from my hand-drawn lecture, is that the cardiac cycle is a repeating pattern of contraction and relaxation of the heart. I want you to understand that the atria contracts first, followed by the ventricle. So atria first, then ventricles. Atria, ventricles, atria, ventricles. Atrial systole, ventricular systole. Atrial systole, ventricular systole. So that's a pattern by which the heart contracts. Two very important definitions here: end diastolic volume, also known as EDV, and end systolic volume, also known as ESV. Very, very important. You must know these two. So right before the ventricles undergo systole, what happens? So the ventricles have not started to contract yet, right? So at that point, do you think there's a lot of pressure in the ventricles due to blood? Yes. So listen carefully. The total volume of blood in the ventricles right before systole is called end diastolic volume. I repeat, the total volume of blood in the ventricles at the end of diastole, and right before the beginning of systole is called end diastolic volume. And systolic volume is the amount of blood left in the ventricle, specifically left ventricle, after systole. So the ventricles have undergone systole, right? So it has contracted, it has pumped out blood. So obviously now there is very little amount of blood left in the left ventricle. And that volume of blood that is left after systole is called end systolic volume, which is about 1/3 of the end diastolic volume. So, easy way to remember: end diastolic volume is before ventricular systole, and end systolic volume is after ventricular systole. Okay, and obviously this is 1/3 of EDV. Sorry, this should be ESV. I'm sorry. Okay. All right, moving on to the conducting system of the heart. And I love to draw this out on the whiteboard, and I'm going to do that on my surface to give you a feel of the conducting system of the heart. So let's get started. Conducting system of the heart. So this is the human heart. This is how I like to draw the heart here. And the human heart has 4 chambers. So let's draw them. And let's label the 4 chambers. This is right atrium, left atrium, right ventricle, and left ventricle. Separating the atria and the ventricle, we see a thick structure, and this is called the fibrous skeleton. The fibrous skeleton, it acts as an insulator. Very important. This acts as an insulator between the atria and the ventricles. All right. Now, there are few things that you must remember, and let me do it this way so I can use different colors. Okay, so there are two to start with. There are 2 types of cells that we will talk about. First, we have the myocardial cells. And the myocardial cells, these are nothing but the cells of the heart muscle. And then there are some specialized cells. These are very, very specialized. They are not myocardial cells. And these specialized cells, they actually generate the heartbeat. So these specialized cells, they generate heartbeat. Okay. And since the heartbeat is generated by the specialized cells without any innervation from the nervous system, That's why our heart is called autorhythmic. Autorhythmic. So the human heart is autorhythmic, meaning it can generate its own beat without any influence from the nervous system. All right. So where are these specialized cells? Well, we have these specialized cells in groups or patches. And first, we have in the posterior superior part of the right atrium, we have a patch of these specialized cells, and this forms our sinoatrial node, also known as SA node, also known as pacemaker of the heart. Okay, the SA node, so that's the SA node. Then we have the AV node, which sits right here. This is called the AV node or atrioventricular node. The AV node then is connected to groups of other specialized cells, and this is called AV bundle. The AV bundle comes down into the ventricle and then splits into two branches. We have a right bundle, and we have left bundle, and they terminate in these fibers, and these fibers are called Purkinje fibers. So let's review the anatomy one more time. We see 4 chambers of the heart, and the atria and the ventricles are separated by fibrous skeleton. Obviously in the heart we will see myocardial cells, and remember we have about 99% of the cells are actually myocardial cells, and then there are specialized cells which are about 1%, and the specialized cells, they can generate the heartbeat, and that's why the heart is called autorhythmic, meaning that it can generate its own beat without innervation of the nervous system. We do see these SA nodes on the superior posterior side of the right atrium. We have the AV node on the inferior side of the right atrium. The AV node leads to AV bundle. The AV bundle then moves towards the ventricle and then divides into left and right bundles, which ultimately terminate in Purkinje fibers. Good. Before we start talking about the electrical conduction, how the electricity is generated and conducted, I want you to remember one thing. Always remember, electrical events precede mechanical action. So you will always have electrical event first, followed by mechanical contraction, right? We see the same pattern here. All right, so let me take a different color. Okay, so first what happens is the SA node starts firing some action potential. SA node starts firing action potential, and these action potentials spread all over the atria and depolarize the atria. Right? These action potentials then hit the fibrous skeleton, but remember the fibrous skeleton is the insulator, so most of these action potential dies down. The only point that holds the action potential is the AV node. The AV node delays the action potential for about 0.1 to 0.2 seconds, and then it lets the action potential travel down into the ventricle via the AV bundle. So this is the pathway of the flow of action potential. It comes down to the ventricles via AV bundle, then moves along the left and right bundle, and then ultimately moves to the Purkinje fibers. This is how the action potential reaches the ventricle. Good. Now, right after the action potential spreads all over the atria, we will have a mechanical event. So we will have atrial systole. And the AV node delays the signal, right? So after 0.1 to 0.2 seconds, the AV node helps the action potential to move down to the ventricles. And then after the entire ventricle gets the action potential, we will have ventricular systole. So remember, we have atrial systole followed by ventricular systole. All right. Now, another thing. The SA node cells, the AV node cells, the AV bundle cells, the right bundle, left bundle, Purkinje fibers. Remember, these are all specialized cells. These are all specialized cells. Now, these cells are connected to each other by gap junctions. Remember gap junctions? So, these cells are connected to each other by gap junctions. Therefore, through these gap junctions, the action potential can flow really, really fast. So let me write this down. So the cells, the specialized cells, are connected to each other by gap junctions. Now, are these specialized cells connected to regular myocardial cells as well? Yes. These cells are also connected to myocardial cells by gap junctions. Therefore, please hear me. The action potential that are generated by the SA node, they spread all over the atria because these specialized cells are connected to the myocardial cells by gap junctions, right? So the wave of depolarization can spread from cell to cell to cell to cell to cell all over the atria. Good. So remember that the specialized cells are connected to each other by gap junctions, and these specialized cells are also connected to myocardial cells, and each myocardial cell is connected to other myocardial cells by gap junctions. All right. Moving on to the next slide. So looking at the electrical activity of the heart, as I mentioned before, the cardiac muscle cells are interconnected by gap junctions, and the gap junctions are located in special areas called intercalated discs. Now, once a stimulus is applied, the impulse flows from cell to cell to cell to cell to cell. And as the impulse flows super quick, or as the action potential or the wave of depolarization runs from one cell to the next cell to the next cell to the next cell, all the heart cells at a particular area contract together, and that's called a functional syncytium. Another important thing to remember, the atria and the ventricles are separated by the fibrous skeleton, as we mentioned before. So this is a nice little summary slide for the electrical conduction of the heart. Now this is another picture showing electrical conducting system. So let's take a look at the picture first. On top right here, this is the SA node. The SA node starts firing action potential, which spreads all over the atria via gap junctions, and that helps the atria to depolarize. The action potential then hits a fibrous skeleton, which is located right here, but most of the action potential dies down except for the AV node. The AV node holds a signal for 0.1 to 0.2 seconds and lets the signal flow through the AV bundle into the ventricle, and the signal then divides, and the signal then travels along the left and the right bundles and then ultimately moves to the Purkinje fibers. And this is how the wave of depolarization starting from the AV bundle spreads all over the ventricle. Atrial depolarization is followed by atrial systole, and ventricular depolarization is followed by ventricular systole. I'll stop here and then start